Hi there. Today's video is going to be a really quick RNA-seq analysis comparing Crohn's disease, ulcerative colitis, and healthy colon tissue. I pulled the data from today's video from the published data sets in Pluto. So I just entered the keyword IBD and it returned 45 results containing IBD from the 15,000 published experiments available. So I selected the data set that I was interested in, and you can see in this published data set, we have the curated sample metadata from all of our samples, as well as the assay data. So that includes the raw counts data, as well as the FASTQ files. I wanted to basically start from scratch and bring this over to my own project. So I just copied that to my IBD immune project. And then we can head over there to begin. Okay, so you can see in this copied project that I have that sample metadata and I have the assay data, which is raw counts. That's really all I need to begin an analysis on Pluto. So I can head over to Analysis Grid, and you can see I've already created all of these visualizations, and I'll walk through how I did that. So I click on Add Analysis. And first, I want to compare gene expression in the Crohn's colon relative to the healthy colon. So I'm going to create that analysis by basically combining two variables of disease state and tissue. And again, this is pulled from the sample metadata. So I can select my experimental group as the Crohn's disease colon and the control group as the healthy colon, and then run analysis. So that will create a nice volcano plot for us. And then what I want to do is explore a few genes of interest. So this paper, this data set was used in a paper from July where they identified genes that were drivers of immune infiltration and tumor progression in IBD-associated colorectal cancer. So I just pulled a few of the genes that the authors identified as key genes in the abstract. And then to specifically look at those genes, I went down to the labels section under plot. And I could label an entire list that I had personally curated or a list from IMSIGDB, but I want to search for those specific genes from the publication. So I just clicked on those 4 targets and saved changes, and then it colors and labels those 4 genes of interest for me. So you can see in my kind of finished plot that I've also gone ahead and changed the colors and added a p-value line to just make it really clear. So I did that same exact analysis in the ulcerative colitis colon relative to the healthy colon and identified that, again, those 4 key genes from the publication were also upregulated in the ulcerative colitis condition relative to healthy. There is another way to kind of examine that question. So rather than looking at log-fold change as we do in differential expression, we can also just do a summary analysis on the normalized counts and compare the normalized counts. So that's what I've done here on the left-hand side. I've just entered those 4 target genes and grouped by disease state and tissue. And again, I see that those 4 genes are more highly expressed in the Crohn's disease colon compared to the healthy colon. I also ran 2 sample t-tests comparing those groups for each of the genes, and we see that indeed their expression is significantly different. Another analysis that we can do on Pluto that's very common for RNA-seq is gene set enrichment. So let's actually run one of those. You can click on gene set enrichment and create analysis and select our Crohn's disease versus healthy colon comparison. And then we will choose a collection from IMSIGDB, but we could also build a custom gene set collection. So let's select the hallmarks and run analysis. So this is actually running GSCA on every single gene list that is available in the Hallmarks collection. And the first thing that it returns to us is the most significantly enriched gene set, which in this case is the epithelial mesenchymal transition. But we can also go in and look more specifically. So we can expand that and see all of the gene sets that are a part of the Hallmark collection. So let's select the inflammatory response gene set and save changes. And then we see that GSCA plot change to reflect the inflammatory response. So we do see this kind of peak on the Crohn's disease side that affirms that inflammatory response genes, or genes that are a part of that collection, are more highly expressed in the Crohn's disease colon. So finally, I want to run a comparative analysis where I actually overlap the genes that are upregulated in the Crohn's condition and the genes that are upregulated in the ulcerative colitis condition. So let's run one more analysis, and we can do a Venn diagram overlapping our genes. So I'm going to create a list of differential targets, and we'll call this Crohn's up, and select all the genes that are upregulated in the Crohn's condition. And then we can select ulcerative colitis up and select all the genes that are upregulated in the ulcerative colitis condition. Save that, and we get our nice Venn diagram overlapping those conditions to identify the kind of shared set of genes that are upregulated in both. So what we walked through today was pulling in a published data set from Pluto into our own project, running a few analyses on that, like differential expression, gene set enrichment, summary plots, and a Venn diagram, to identify if certain key genes that the authors had indicated in this paper of interest were indeed upregulated in the Crohn's condition and the ulcerative colitis condition. I think what's really cool about this is that typically pulling in a published data set can take, days to go out to SRA and fetch the FASTQ files, or go to Geo, and sometimes the counts data is, not very meticulously curated. So this is just a really fast way to grab a public data set, compare it to the author's findings, and confirm or deny what they had identified in the publication. I hope this was helpful, and I'll see you in the next video. Bye.